Hello everybody. Today I will explain my ideas over the cell scanner. Although such a treatment will not exist for a long time, it might be required to conquer cancer and other diseases caused by somatic mutations in an effective way, and even slow down aging. Therefore, it is advisable to start the proper scientific research already now. During this presentation, we discuss the following topics. Surgery, like removing the appendix, treatment of the pituitary and plastic surgery, eventually supported by cameras and robots, are probably the most spectacular treatments of the current medicine. When possible, even a less incriminating keyhole surgery procedure is executed. Also various fractures, among which fractures of the arm and leg, can currently be treated very well. Other macro interventions, like bypasses, organ transplantations, and the removal of tumors are today common practice. Bacterial infections are today treated with antibiotics, initiated with penicillin by the Scottish doctor Alexander Fleming in 1928. Such a medicine remains until today effective for killing bacteria. Currently, however, an increased resistance against the usual antibiotics is problematic. Viral infections are more difficult to cure. Most of them can be prevented with an in-advance injection of the appropriate vaccine. This much improved method was invented in 1796 by the British Dr. Edward Jenner while he was experimenting with smallpox. Some diseases caused by a virus or prion, like Ebola, HIV and the disease of Creutzfeldt-Jakob, are still life-endangering. The current medicine is however mostly based on a designed molecule, and is developed by pharmaceutical companies. It is often taken as a pill, tablet, capsule or syrup. Sometimes, it is injected or administered with a Baxter. New medication, before it can be used in the European community and North America, has to be approved by the European and American medicine authorities. Such an acceptance is based on positive evaluations of a set of clinical trials, in which the medicine is tested on humans. These are executed after successful preclinical studies and guarantee safety for the person. Also, the effectivity for the ALS person is investigated by comparing sick and healthy participants. The right dosage is also determined. Yet a severe regulation contains a crucial limitation. Treatments that may prevent or delay diseases when there are still no visual symptoms cannot be decently tested. Perhaps a similar approach as for the approval of vaccines could be applied, in which only the expected effect is achieved before one is ill, instead of recovery for one that is already ill. Despite a large improvement the last century, there are still a lot of diseases which are difficult to cure or even incurable. Plenty of cancers still end badly, and for a lot of neurodegenerative diseases like Alzheimer, ALS and Parkinson, there is no cure. In addition, many autoimmune diseases, such as the disease of Crohn and multiple sclerosis and disorders caused by a virus or prion, like HIV and the disease of Creutzfeldt-Jakob, are still waiting for a curable treatment. This makes us aware that medicine has not reached its endpoint by far. Most medication uses the digestion system and blood circulation and reaches, beside the targeted tissues, also other cells. On top of that, a lot of medication is not selective enough for solving the intended problem. Therefore, current medication may have malicious side effects. In addition, looking only at pharmaceutical means is often inadequate. Today, we can speak of macro medicine. The greatest progression can probably be achieved in the internally Directed manipulation of cells. Intervening in the metabolism of individual cells is most likely the principal challenge of the medicine of the future. This involves technology that deals with very large numbers. Examples of this are approximately 4 times 10 to the 13th power cells in the human body, about 10 billion proteins and 3 billion base pairs in each cell. Also, medication of the future will only affect the correct cell types. A futuristic cell scanner must be able to reach every cell, as each cell requires oxygen. The blood circulation system that supplies all cells could be followed. The cell scanner could therefore best directly be injected in a blood vessel. For sure, one has to take in account the blood-brain barrier where astrocytes prevent direct contact between nerve cells and the blood circuit. Treating 4 times 10 to the 13th power cells in one cell scanner session is probably too much to ask. By only looking at the target zone, this number can be drastically decreased, but the amount of cells to pass by remains considerable. Also, a self-destruction mechanism will be required. Time is further of the essence. A treatment may last a few hours but surely not days. Additionally, cells, being about 10 micrometer tall, are very small. The use of nanobots, with a size range of 0.1 to 10 micrometer, might solve this problem. Not only should all cells be located in the targeted area and indicate that they are already scanned during this session, they should also be identified. First, body cells should be distinguished from bacteria. Furthermore, the difference between a normal and stem cell should also be determined. Finally, the found cell has to be harmful before the appropriate action, destroy or repair, is executed. After identifying the scanned cell, action has eventually to take place. Destroying harmful cells is probably the simplest solution and will often be sufficient. However, sometimes a cell correction, like repair of the telomeres, the removal of protein remains, and the correction of DNA encoding, a more suitable option. The cell scanner must also report its activities. Useful information is when something becomes active, what is found, which action has been performed, and when self-destruction has taken place. A lot of diseases are probably caused by certain local somatic mutations in genes of a stem cell during lifetime cell renewal. It is naive to think that somatic mutations only occur in cancer. Some mutations also might cause autoimmune diseases, like MS and Crohn's disease, and neurodegenerative illnesses like Alzheimer, ALS and Parkinson. All these diseases have in common that they are caused by damaging somatic mutations that slip through the net of the DNA control during the process of cell replacement. The cell scanner can eliminate or repair cells with harmful mutations and stop the progress of such disorders. Also, viral and prion-related diseases, like HIV and the disease of Creutzfeldt-Jakob, could be handled with such a treatment. New cells are made during life using stem cells. For that, DNA is copied and copy errors, named mutations, are sporadically made. Also, damaging protein remains accumulate in cells, like most neurons, that are not renewed. Aging is the consequence of this and can be retarded by directed cell scanner sessions. Neurodegenerative diseases, like Alzheimer, ALS and Parkinson, can no longer be cured by repairing mutations but can probably be prevented with early cell scanner sessions correcting stem cells for nerve and nerve supporting cells. The development of curing cell scanners will not happen in the near future, and I will surely not be able to make use of them. Still, a lot of questions, like these shown here, require an answer. This, however, does not mean that we cannot start now with the proper research for this. In such research, Multiple disciplines have to cooperate. The multidisciplinary group Science and Technology of the KUL, headed by Vice Rector Gerard Govers, offers maybe the right platform for this pilot project. This will however require the addition of other faculties like medicine and biology. Also, the company IMEC is a good candidate for this. Engineers can focus on the finding and identification of cells. Physicians and biologists may concentrate on the destruction and reparation of cells. A bioengineer may coordinate their activities, especially as useful know-how, like gene technology, under the wing of Professor Rob Levine, and biosensors, directed by Professor Jaron Lamertin, are part of the Faculty Bioengineering Sciences of the KU Leuven.